Thank you very much for the invitation. It's been a fun conference, and thank you for sticking it out to Wednesday morning at 8 a.m. So today what I want to tell you about is work in my lab, and my lab essentially uses genomic tools and ancient DNA methods to answer anthropological questions about population history in humans and non-human primates. And part of this is also thinking about the pathogens that have affected us as well as other animals and that exchange. So the research questions that we have been focusing on in the context of one major pathogen, Mycobacterium tuberculosis, you can see here. Now, we're also in my lab studying some other mycobacteria as well as some other pathogens, but I'm going to focus on this story today. And in particular, we've been interested in what the origins of tuberculosis are in humans and what the pattern of strain diversity is kind of pre and post age of exploration, age of colonization. Because of course we know during that time period many people came in contact with each other that hadn't been in contact previously. And also, of course, they are moving domesticated animals and plants around the world during this period. So it's a very interesting period of time. And within that, my laboratory has focused on the question of tuberculosis presence or absence, or exactly what it was in the Americas. So were pre-contact strains in the Americas, what were they? Was it even there? And then post-contact today, tuberculosis strains in the Americas are basically all European strains. So when and what was the pace of that replacement or introduction of strains? So when you work with the evolutionary history of infectious diseases, you very quickly gain a One Health perspective and start thinking about the exchange among humans and other animals, as well as among animals in general. And so we quickly became interested in that in the context of mycobacteria here. When you use ancient DNA, you also have a time component, right? And so we can also ask the question, how have mycobacteria adapted over time? So a little bit of background first about mycobacteria and tuberculosis, and then a bit about ancient DNA. But Mycobacterium tuberculosis is part of the genus Mycobacterium. And this is a large genus that is mostly harmless soil and water saprophytes. And I bring this up because when you're working in an archaeological context, you often encounter soil and water, and it comes back to haunt you later. So you'll hear about that. But many mycobacteria are opportunistic pathogens. My personal favorite is Mycobacterium jacuzzi. Guess how that got found? Mycobacterium tuberculosis itself is the primary causative agent of tuberculosis in humans, but it's part of a complex. And this is where you get into what is a species in bacteria, but you could call these species, subspecies, or ecotypes. But basically, you can see this constellation of species, listed here as species, actually, M. africanum, M. bovis, M. cannedii, M. microti, etc. And all of these can infect humans, and all of them cause the disease known as tuberculosis, and all are treated in the same way. In addition to humans, these affect our domesticates, and they're typically, these names that you see here are typically based on the species in which the strain was discovered. However, there's actually a very poor understanding of the host range of each of these, with the exception of M. tuberculosis, which is pretty clearly human. It's fairly rare for other animals to get M. tuberculosis. So that one seems to have really focused on humans. But the others, M. bovis, it's found in a range of livestock. It's also found in other animals, wildlife as well. And so exactly what the original host range or the original, the array of species that are affected is not very well understood, in fact. If we look at the genome data that are available as of 2017 in this phylogeny of mycobacteria, what you can notice is that most of our genome sequences, not surprising, are from the slow-growing group of mycobacteria. So let me see if I can use the pointer here. Oh, evidently I cannot use the pointer on this. Okay. So if you look on the screen and you see the growth rate, you can see rapid and slow, and most of these are in the kind of the blue slow. The faster ones tend to be the environmental. Some of those have been discovered because they've infected immune-compromised individuals. And you can also see Mycobacterium tuberculosis, and that particular group is, you're trying to tell me to use this, aren't you? I was trying to avoid this because I know everybody in the back can't necessarily see it. But at any rate, in gray, what you can see are the Mycobacterium tuberculosis complex, so that group of so-called species or ecotypes. And they're very, very closely related. Okay, so what is tuberculosis? Typically, it's an infection of the lungs and inhaled. Very small percentage of people who are exposed actually become ill. Many people actually wall off the TB into a granuloma. You can see that in this chest x-ray. And there it stays. And maybe later, if they become co-infected with something else, or they, as they age and the immune system declines, it can basically break out of jail. Now, you can also acquire tuberculosis through the gastrointestinal route, and prior to the pasteurization of milk routinely, that was a major cause of infection or route of infection for humans. This quote here sort of nicely summarizes the symptoms. This is actually from the USDA fact sheet for M. bovis. So when we look at other species, the characteristic symptoms are the same in essence. Tuberculosis in humans is now currently the number one in terms of cause of death by a single infectious pathogen. It has gotten in front of HIV, and it's actually the leading cause of death in people with HIV. You can also see here from the World Organization for Animal Health, unfortunately we don't have the same level of statistics, but in countries that reported, 44% reported the presence of M. bovis. So we know that that's a major of major importance for agriculture in the sense of affecting productivity and health in animals. So what do we know about the earliest cases of TB? Well, the first clear historical evidence is from China. This is a description where everybody can agree that, yes, that's actually tuberculosis. We do have skeletal evidence in the archaeological record. So tuberculosis, unlike many infectious diseases, if you have a chronic case and you hang around for a long time, it can disseminate into your bones, and it causes these sort of holes that you see here in this x-ray, and it can cause the vertebral body to collapse, resulting in something known as Pott's disease. And we see depictions of what could be TB in art. However, to be honest, if somebody had tuberculous Pott's disease, I seriously doubt they'd be irrigating. I don't think they'd be feeling so well, at any rate. So we do see tuberculosis in the archaeological record. However, the archaeological record for other species regarding tuberculosis is nil. So not surprisingly, animals that develop chronic TB usually are culled or predated. And so we just don't see these lesions in animals in the archaeological record. So what about the evolutionary history of tuberculosis? The early thought was that M. tuberculosis evolved from M. bovis in the old world with cattle domestication. And so you might think, but hey, you work in the Americas mostly. And yes, that's true. And the Americas were colonized roughly 15 to 20,000 years ago, give or take. We have various models about the number of people that were part of this migration. We're pretty clear on the number of cows, 0. So as a result, if M. bovis is the progenitor of M. tuberculosis, then we shouldn't expect Mycobacterium tuberculosis or parts of the complex in the Americas before contact. Well, archaeologists can be pesky, but they do really interesting work and provide the context for a lot of the genetic work that we do. And so I collaborate with Jane Bijkstra, who is a bioarchaeologist and for many, many years has studied the bioarchaeology of the Americas, and in particular has been interested in tuberculosis. And over the course of her career, she, and as well as others, increasingly found evidence for classic tuberculosis, so these lesions that you see in the spine and elsewhere in the body. And in 1994, they analyzed DNA from a mummy from Peru that had TB-infected nodules. And this work was published in PNAS, and this was the height of ancient DNA research in 1994. They got a band. It was great. Okay. I wish I could get a band and get a PNAS paper. So there we go. They did the sequence. It's a sequence from IS6110, which is a repeat element. And unfortunately, all IS6110 repeat elements are basically the same. So basically, what they could say is TB appears to be present, since IS6110 is pretty much only found in the TB complex. So it's kind of a yes-no marker. And they couldn't really put it in larger context. And to be honest, at the time, there really wasn't the available data from other strains of mycobacteria from around the world to compare with, even if had they gotten additional data. But starting in the late 1990s and early 2000s, we do start getting the data first as insertion deletions, these RDs, regions of difference, as well as a few SNPs. And these showed us pretty clearly that M. bovis is more derived compared to Mycobacterium tuberculosis. And we see this in genome data. This is from just last year. But what you can see pretty clearly, and these colorful clusters are the human groupings of Mycobacterium tuberculosis, and then interspersed among them and more derived are the animal strains. And so it's pretty clear that cows did not give us tuberculosis. We may have given it to cows. Poor cows. Okay. So if you look today at the biogeography of tuberculosis, what you see is that this is basically, by the way, these colors that you see, I'll show over and over throughout the talk. They'll be consistent. But in essence, when you look at this phylogeny that was published by Gagnon et al. And Hirschberg et al., what you see are strains that share a deletion that seem to be spreading the most today. They propose that these might perhaps be more virulent. This includes the Europe and American strains that you see here in red. And then these older strains are found in Africa as well as around the rim of the Indian Ocean. The animal strains tend to be nested in that group there. And so when we started thinking about what was going on pre-contact versus post-contact, it looks pretty clear that the European strains came in and replaced whatever was there before. But what was there before? And so our original hypothesis, and for those of you who are trainees, I would say this is a fabulous example of a really cool hypothesis that was completely and utterly wrong. Okay, and actually that was even more interesting. So our original hypothesis was that these pink strains that are part of those older lineages came with people across the Bering Strait into the Americas. Okay, but when we started our work, we had to think about other possibilities as well. And so initially when we were designing our experiments, we took that in mind to design our array-based baits, which I'll get into here in a minute. Now, one other challenge that I want to bring up is when you're dealing with mycobacteria, and you're interested in the evolutionary history, well, of mycobacteria, really any bacteria, you have the problem of how do you calibrate the molecular clock. And in mammals and other vertebrates, you have this nice fossil record, and we have hypotheses about when different events occurred. But that's not true so much for mycobacteria. And so when you look at previous work thinking about the molecular clock and the origins of TB, you get this huge range of dates from like 20,000 years to 3 million years that you can drive a truck through. And that's one of the really nice things actually about ancient DNA. And I'm just going to move on here. So initially when we started, we had, when you think about bacteria, you have a couple of events in the fossil record that are recorded, but these are super, super old. And if you use those, of course, it biases your dates backwards. And then we have exciting geologic events like someone threw the type strain into a freezer in 1905, and that's super recent. So that can also bias our dates in different ways. So that's one of the big challenges that ancient DNA can address. And also the when impacts potentially the where this might have happened. Okay, I want to give you a little bit of background on ancient pathogen DNA, since I don't think Eski did. So how do you get at, in particular, pathogen ancient DNA? So we work in a class 10,000 clean room to basically try to protect the sample from ourselves. We take bone samples. Now, a lot of work has focused on ancient pathogens, has focused on teeth, in the case of Yersinia pestis, for example, and leprosy. But one of the things you have to do when working with a pathogen is think about where the pathogen might actually be in the host. And tuberculosis, we so far have not recovered from teeth. And we seem to get the best results by sampling near one of those lesions. So that is what we try to do. The vast majority of DNA in the sample is actually not from the host and not from the pathogen. So a lot of environmental DNA that we have to kind of search through. So previous ancient DNA studies had focused, as I mentioned, on that IS 6110 repeat element. Some people had tried a medical epidemiological way of typing strains, which was spoliga typing. But that's a case where you're looking for the presence and absence of repeats. And the problem with ancient DNA is you have a lot of absence. And that has more to do with preservation than whether or not it's really there. So that's problematic. And so we are often very concerned with DNA preservation and also issues of contamination. So the DNA we're dealing with is these are electron micrographs, and this is very small bits of DNA that's shredded up, so we get to deal with that. And it's often chemically modified, so sort of tied up in knots, as it were. We have samples from probably over, I've kind of lost count. I need to go assign somebody to figure out where we are, but I would say it's over 300 samples with these characteristic lesions of TB, okay? So we're kind of starting with people where we know they had TB. It's a much smaller percent that we can get the genome out of. The first thing we do is a quick set of qPCRs. One is this IS 6110 element that's present in 0 to 27 copies. We also have IS 1081, which is present in 5 copies in all members of the complex. And we also target a specific fragment of a single copy gene, RPOB. So that's basically, can we detect the DNA? Yes, no. And you can see here, what we tend to find is the ones that where we get a yes for all of them are kind of our best bets. But we often typically try all the ones that at least had one positive or two. The next step is to pull out the genome of the pathogen. And we do this with DNA capture and sequencing. So we make a library of all the DNA. Of course, only a small percentage of it's actually what we're interested in. So we use baits that are biotinylated. These focus on, or these anneal with complementary sequences. We attach them to magnetic beads, and we can use that to separate these out from the rest of the DNA. So this is what we focus on for sequencing and subsequent analyses. Now, with ancient DNA analyses, just a little bit about it in particular, up here at the top you can see, this is work by Jesse Dabney a few years ago, but what you can see is a clear indication of the fact that the DNA tends to be highly fragmented. And until recently, and until DNA extraction methods were improved, most of this we actually lost because they didn't retain those short fragments. So that's one thing that we really have to pay attention to with ancient DNA work. Are we recovering those small fragments? And then, of course, you reach kind of a limit. It's usually less than about 25 base pairs where you just can't really accurately map it. And so you sort of lose that. So it's sort of the sweet spot between 25 or 30 base pairs and, say, 60 or 70 base pairs where most of our information is actually recovered. The other thing that we have to contend with is DNA damage, and there's a number of ways to deal with this. But basically, when the DNA is shredded, the single-stranded ends get deaminated. You get what appear to be C to T transitions, and you have to count for that in your analyses. Other work has also looked at the pattern of fragmentation and where it's most likely to occur. So we first focused on 3 samples from 3 archaeological sites in southern Peru. And these are all in the Osmori River drainage. The sites are Chirbay Alta, El Algodonal, and El Ural at various distances from the coast. This region is the northern extent of the Atacama Desert. And so here you can see the river. This is actually avocado orchards today, but the burial sites tended to be up here on the bluff, and you can see just how dry it is, very, very few plants at all. Whereas obviously they were doing their farming then, as we do now, down next to the river. And here's the distances, or excuse me, the dates. So roughly these are all about 800 to 1,000 years old. The preservation there is amazing. So what they would do when someone was buried in the community is they would be placed in a seated position, wrapped in textiles, surrounded by burial goods, carried to the cemetery on wooden litters, which are preserved, as are the textiles, which are amazing if you ever go to Peru. And buried in stone-lined tombs that were then covered with stone capstones. And so here they are in the northern Atacama Desert in a stone-lined tomb, wrapped in textiles, very well protected from soil and water. Okay, so what did we get? And I'll come back to that point in a minute. We were able to recover 3 genome sequences at 20 to 31x coverage. And this is work that was done by Kirsty Boss, who was a postdoc at the time in Johannes Krause's lab, and my graduate student, Kelly Harkins, who's now head of Claret, who had a booth earlier. Okay, so what were our results? Our results were super surprising. So the first major feature that you'll notice here is that ancient Peruvian TB was most closely related to M. pinnipedi. Now, initially when we got these results, it was surprising, and there was also only one M. pinnipedi genome sequence in the database. And this was a South American seal in a German zoo, and that did not inspire confidence. So we spent about a year trying to get basically access to, or even find, mycobacteria that had been sampled from wild M. pinnipedi. And we're finally able to do that, and this is what we found. Now, using these archaeological dates, which are very good, the chronology in the region is very well established, we were able to calibrate the molecular clock, and that led to the other big surprise, which was that it pointed to an origin of extant strains of TB in the last 3 to 5 or 6,000 years. And that was much, much more recent than expected. So it put forth sort of this scenario where TB probably arose in Africa, and this is based just on the fact that that's where we see the most strain diversity today, both in human and other animals. We have no idea where or from what. You then get the spread of these ancient lineages that I mentioned before, followed by a jump back into animals somewhere. I'm putting this here in South Africa for convenience. We get the origin of lineage 7 and the other modern lineages. That one's sort of a progenitor. And at some point, the animal strains jump into probably Cape fur seals. Cape fur seals interact with other species of pinnipeds around Antarctica and ultimately probably pass to the South American pinnipeds, probably 1,000 to 1,300 years ago, or possibly slightly earlier, and then jumped into humans. The next question, though, is did it spread throughout the Americas? You know, this is one of these projects where you find yourself learning a lot about something you didn't expect to, and in my case, this was seals. So I got to learn a lot about seals and stare at Antarctica. Although unfortunately I haven't actually been to Antarctica to stare at Antarctica, but I stared at maps and species distribution of seals. So these are the Cape fur seals, these are the Peruvian fur seals, or the South American fur seals. This is a moche pot from coastal Peru, and it's depicting a seal hunt. So probably the means of transmission was through butchering or eating infected meat that was undercooked. So this, of course, leads to a whole new set of questions. Were there multiple jumps? Does the pathogen readapt to humans, and can we see that in the genome? Does it spread via human-to-human contact into North America? And do other strains of TB come into the Americas prior to 1492? We know that the pesky Vikings visited the east coast of Canada. However, from the archaeological record, we also know that TB was present in North America prior to that time. We also know 4,000 to 5,000 years ago, you get the expansion of Inuit and Aleut ancestors around Beringia or formerly Beringia and into the north, and they potentially could have brought other strains as well. So the other aspect, of course, is what is the pace and source of the spread of TB strains after Columbus? So that's another question. So then we were all excited after getting the first 3 genomes. We thought, great. And then we ran into the ugly truth, which is that most archaeological sites are not in the northern Atacama Desert, and people are not buried nicely wrapped in textiles in stone-lined tombs, but instead interred into the ground in soil where there's often a lot of water. And so our initial results resulted in, or showed us that what we were recovering in the majority was environmental mycobacteria. So we kind of had to go back and redesign our in-solution hybridization to be more specific, and also think about the bioinformatic methods we were using to be able to sort through and distinguish among the species. So in essence, how do we find that needle in the haystack? So where are we now? I'm going to tell you a little bit about some work that is either submitted or in various stages of progress. So Ash Vajane, who was a graduate student at the Max Planck for the Science of Human History, and Tanvi Honap, who was one of my graduate students, worked on a number of samples from around the Americas, and I'll tell you about a few where we do either have genomes or partial genomes. So here, at the first one you see is from Moquegua. That's in that exact same valley, but what's interesting about this particular individual is he was buried close to the river, and so we see a lot of environmental mycobacteria because, and we also see archaeological evidence in the tomb that it was flooded. So not surprising that there's more there. There's also 2 sites in present-day Bogota, La Candelaria Nueva and Las Delicias. Post-contact, let's see, right. Post-contact is Highland Park and Yukon River Valley in Alaska. All right, so first to just tell you about the South American Bogota and Moquegua samples. We have been able to recover genome sequences from those. They're a bit messier because of the environmental contamination. It's kind of hard to pull it out. But when you look at the phylogenetic results from this, they still cluster with the pinniped strains here. So these are the, these are the original genomes, and these are the new ones. So all of it's clustering with TB. With M. pinnipedi. One of the early criticisms of our first paper was maybe these were just zoonotic TB. Because the River Valley, 2 of the sites show clear interaction with people on the coast, or fishing and things like that, so clear use of marine resources. But when you move to looking at a site like the 2 in Bogota, there's pretty clear evidence that they weren't eating seals. There's not really seals at 2,000 feet up in the Andes. So I think it's pretty clear that that's probably via human-to-human transmission. We also, in looking at radiocarbon and isotopic data, show that these weren't people who were living along the coast who went up to the highlands later in life, from what we can tell. Okay, so what kind of changes might we see in these genomes? We do see quite a number of non-coding SNPs, and we've tried to think about, do any of these show evidence for adaptation? We're also, quite a few are non-synonymous, and many of these have multiple non-synonymous, many genes have multiple non-synonymous SNPs. These are in a range of genes, some of which do seem to perhaps help the pathogen adapt to the host. But it's kind of unclear, and dealing with the population genetics of a clonal organism that has sort of boom-bust population dynamics is quite tricky. So I'm actually also working with some theoretical population geneticists, in particular Jeff Jensen, to think about some of these dynamics and how they affect what we can infer about not only timing, but also selection. So we have additional ancient TB genome captures in progress, and one of these is, or we have some preliminary work from my graduate student who's super happy about it, Kelly Blevins. And she just spent a year on a Fulbright to Mexico, where she had a wonderful experience working in the collections at the National Museum. And what's really exciting is she's focused on individuals who were buried at the Aztec market of Tlatelolco. So Tlatelolco was established in the 1300s. Of course, contact occurred when Cortez showed up, but this was one of the largest, if not the largest market in the world at the time. It's estimated that 50 to 100,000 people would show up at this market every day, and she found quite a few cases looking through the paleopathological record of what looked like tuberculosis. And I think this will be super exciting because when you have such a large market and people coming from all over, we would expect a fairly high diversity of strains. So currently what we have are preliminary data from 2 strains that she's in the process of analyzing. The one thing I will tell you is that, or I'll tell you 2 things. Our initial analyses show that these are M. pinnipedi. So M. pinnipedi, through trade networks, made its way from South America up into at least Central America and Mexico, North America. So 2 things I forgot to say previously. When you look at the distribution of M. tuberculosis in pinnipeds, it's only southern hemisphere pinnipeds that have TB. Northern hemisphere pinnipeds, who are very well studied in terms of disease, to some extent I would say, have had no reported cases. The other thing that I forgot to say earlier is that what's really interesting about our results in Peru is that one big puzzle in paleoarchaeology or bioarchaeology would be, why do we see the earliest cases of TB in the Americas in South America if people brought it coming over the strait? And so the answer seems to be people didn't bring it over the strait. It came first to South America. Initially, people thought maybe that was an aspect of preservation or something, but it really does seem to be actually that's where it was first. And now we're showing that human-to-human contact is most likely what helped it move north. One thing that we have done in terms of analyses is look at whether we can see these different deletions, these regions of difference. And what you can see on this particular, you can see the different regions of difference. You can see the different sort of ecotypes or species or whatever you want to call them. And here you have M. pinnipedi, and there's a specific deletion that's called RD seal, and it's most closely related to M. microti, which has its own deletion, and then there's several that go on to M. capra and M. bovis. But in looking at the two strains from, okay, so I pointed at it. All right. So when we look at our data and we compare the Peruvian strains, the Colombian strains, and our data from Mexico, what you can clearly see is the RD seal is present in the Peruvian and Colombian individuals here, but absent in the strains from Mexico. So this also potentially points to variation in the M. pinnipedi that existed at the time and that jumped into humans probably on multiple occasions. We also have some preliminary data from two post-contact contexts. And these, of course, are focusing on this lineage 4, which is the European American lineage. And within that lineage, there are other clusters, and in particular, the two cases that I'm going to tell you about are found in L410 and L4. Good grief, I can't even see that. L42, yeah. So the first is from a poorhouse that was in use from 1826 until almost the end of the Civil War. And what's really fascinating about this is it's from a poorhouse in upstate New York. And if you'll recall, I mentioned H37RV, that strain that was put in the freezer in 1905. That was also from upstate New York. And what you see is that the strain from Highland Park is very closely related to that strain that was thrown in the freezer 80 to 60 years later. Okay, so not surprising. And also what we see is that these are related to other strains that are prevalent in New England and Canada. And if you think about the colonization history of New England, and then it's not surprising that the strains are related to strains that are also very common back in England and in Northern Europe. All these new genomes, this is to remind me to say that they don't change that date of the TMRCA or of the complex. So still 3,000 to 6,000 years. And then finally, we have one from Alaska. And this is the other one I want to talk about briefly. And what's interesting about this one is it's in a different group that's pretty rare, the L42. And this is most common in parts of East Asia and in Russia. And of course, Alaska was first contacted by Russians and whalers and fur trappers. So they seem to have potentially brought strains. Now, the other challenge, though, of course, is that Inuit and Aleut ancestors were interacting across the Bering Strait. So that's another possible mode as well. So what about what's going on in animals and in particular, you know, as domestic animals are being moved around the world, can we see traces there? And unfortunately, we don't have evidence yet in that regard. We've thought about accessing faunal data, but it's a bit daunting. So we decided to start by looking at pinniped populations in Antarctica. And so this is work in collaboration with Mike Goebel, formerly of NOAA and now at UCSC. And he, going there every year for the last few years, has brought us back spit samples from Antarctic fur seals, leopard seals, and Weddell seals. And it's fur seals that we have most of. And you might be wondering, how do you get spit from a seal? And the answer is, I don't know. But they collect a number of different biological samples. And I said, how about spit? And they're like, yeah, okay. So at any rate, this is just to show you the location of the collection. This is from, this box it's very difficult to see, but it's from up here, just across the strait from Tierra del Fuego. And as you can see, this is a temporary base, and the seals are just sort of hanging out. And evidently, they get spit. And unfortunately, I never know how to phrase this. Unfortunately, none of them were sick. Right. But, if you think about a prevalence of 1%, you've got to go through a lot of seal samples to actually catch a case. So, generations of undergrads in my lab get to do the annual seal spit extraction, I think, until we can get some cases, hopefully, to look at the pattern of we don't actually have any strains from Antarctica or know the distribution among species. So to finish up, what are the origins of TB in humans? We still don't know. What's the pattern of TB strain diversity before and after the age of exploration/colonization? That has definitely changed and is something that we still need to do a lot more work regarding. Initially, TB appears to have come into the Americas via seals, which was highly unexpected. But there may have been other routes, and so we're definitely interested in piecing that together. And also piecing together the pace and pattern of introduction of European strains post-contact. Have mycobacteria adapted over time? That's still an open question. My suspicion is, of course, yes, because life tends to do that. But how do we look at those signals in a clonal organism with a boom-bust population dynamic is also a question. I did put those up. Oh, sorry. I thought I had. Okay, there we go. Yay. And then how do strains in other animals relate to the strains found in humans? We do have a lot of data from domestic animals, mostly in bovis genomes. Wildlife is another story. Unfortunately, we need a lot more information about the variety of pathogens, not just viruses, but also bacteria that inhabit other organisms, and so that we can better look at that exchange. And we know that that's important from the One Health perspective, from the human health perspective, and from the perspective of agricultural productivity, because there is exchange. And here you see infected wildlife being represented by a deer, I guess, but badgers and possums in New Zealand, and even pinnipeds have also given tuberculosis to livestock. So, for example, in New Zealand, in the screenings for M. bovis, they've detected aberrant strains, and further analysis has shown over the last 10 years that there have been 9 cases of M. pinnipedi jumping into cattle. When interviewing the farmers have said, I should say farmers, multiple, they have seen seals on the property. And so probably, I guess they're coughing on the grass and then the cows are coming along. I don't know. Maybe they're having little conversations. But at any rate, the seals can really sneeze. These are getting transmitted, and you do see this exchange. But it's kind of rare that we pick these things up, and I think it's important to look further. Finally, of course, I have to thank the many people in my lab and my collaborators, as well as funders. In addition, many museums and First Nations and Native peoples have consented to participation of their ancestors in this research, and that's deeply appreciated. So thank you very much.